So, so what is linkage? Linkage is the tendency of the two genes to stay together. And why do they stay together? Because they are present on the same chromosome. Now please remember, accordingly, genes can be on the same chromosomes or they can be on the different chromosomes also, because it all depends upon which two characters you want to study. Inheritance topic, if you remember, he took combinations of characters also. First he performed monohybrid cross, then he performed dihybrid. So in dihybrid he randomly picked up characters like height of the plant and all. So if you remember, in the last classes I have explained this concept to you. And if you remember, there was a sheet where I explained to you the concept that genes are present on the different chromosome. So luckily, whenever two genes are present on the same chromosome, they will have chances to go together. And the closer the two genes, here three are present, closer the two genes are, more chances to go together. So when they have more chances to go together, they will not separate. That means that something like capital R and small r, this combination will not occur because crossing over will not separate these two genes. That means recombinants won't be formed. That means the 1:1:1:1 ratio will be replaced by 1:1 ratio. So that means genes can be called as two types. If they are present on the same chromosome, they are called as linked genes. And if they are present on the separate chromosomes, they are called as unlinked genes. Now what will happen if it is a linked gene? If it is an unlinked gene, then in the dihybrid cross, the ratio in the F2 generation will come 9:3:3:1. And in the test cross, the ratio will come 1:1:1:1. But if two genes are linked genes, then the dihybrid ratio will not come 9:3:3:1, but it will come as 3:1. Reason, two genes are not separating because they don't separate. Recombinants are not possible because these are the ratios of recombinant, if you remember. And in such a case, instead of 1:1:1:1 ratio, the ratio of the test cross will come 1:1. And now again, one more point, that linkage is also of various type. It can be complete linkage or it can be incomplete linkage. Now what is complete linkage? If I draw a chromosome like this, another one like this, if two genes which you are studying are very close to each other, then you can call them as that they have more degree of linkage, or you can even call it as a complete linkage. And when two genes are far from each other, or maybe this one and this one, on the same chromosome, that means linkage is but presence of gene on the same chromosome, but they have different degrees of linkage. Closer the two genes are, high degree of linkage. Farther the genes are on the same chromosome, lesser degree of linkage. So accordingly, the ratios, the probability of formation of recombinant is going to change. If it is complete linkage, in that case, if it is complete linkage, then the case will be what? This will be the ratio, 1:1 ratio. If it is completely non-linked, it will be 1:1:1:1 ratio. I hope it's getting clear. We'll continue this topic in the next class.